Loneliness is usually described as an emotional experience, a feeling of being disconnected, of wanting more contact than you have, of something missing in your social life. That description is accurate, but it is incomplete, because loneliness is not just a feeling, it is a biological state, one that produces measurable changes in the brain, in the immune system, in the cardiovascular system, and in the hormonal environment. Changes that, when the loneliness persists over time, accumulate into health consequences that most people would not expect from something they would describe as merely an emotional problem. The research on chronic loneliness has produced findings that are striking enough to change how medicine thinks about social connection, not as a lifestyle preference, but as a physiological necessity with consequences for physical health that are comparable in magnitude to smoking, obesity, and physical inactivity. Understanding the biology changes what you do about it, not by making loneliness feel more manageable emotionally, but by making it clear what is actually at stake biologically when social connection is chronically insufficient. The first finding that reframed how researchers think about loneliness came from neuroimaging studies examining what happens in the brain when lonely people are exposed to social exclusion cues. The brain region that activates most strongly in response to social rejection and exclusion is the dorsal anterior cingulate cortex, the same region that activates in response to physical pain. This is not a metaphor. The overlap in neural circuitry between social pain and physical pain is direct and anatomically specific. Social exclusion and a burn to the hand activate the same region of the brain. This overlap has a proposed evolutionary explanation. Social connection was survival critical for most of human evolutionary history. Exclusion from a group meant death. The neural system that monitored for social threats therefore borrowed from the most urgent signaling system available, the pain system, to ensure that social disconnection was treated with the same urgency as physical injury. The consequence is that chronic loneliness is not experienced by the brain as a mild background dissatisfaction. It is experienced as a persistent, low-level pain signal. And persistent pain signals have biological consequences. They activate the stress response, they dysregulate the immune system, and they alter the hormonal environment in ways that accumulate over time. Cortisol is the primary hormonal consequence of chronic loneliness, and the mechanism runs through the threat detection system in a way that is now well documented. The brain of a chronically lonely person is in a state of heightened social vigilance. It is continuously scanning the environment for signs of social threat, rejection, exclusion, hostility, because the evolved function of loneliness is to motivate the search for social connection by making the absence of connection feel threatening. This heightened vigilance activates the HPA axis, the hypothalamic pituitary adrenal system that produces cortisol at a chronically elevated level. Lonely individuals consistently show higher cortisol throughout the day compared to socially connected individuals, even in the absence of any specific stressful event. The stress response is running on the perception of social threat rather than on any specific external stressor. The consequences of chronically elevated cortisol are the same as those discussed in any context of chronic stress: immune suppression, cardiovascular damage, disrupted sleep, muscle catabolism, and impaired prefrontal cortical function. Loneliness does not produce these consequences because it is emotionally difficult. It produces them because the biological response to chronic loneliness is functionally identical to the biological response to chronic threat. A study published in Proceedings of the National Academy of Sciences found that lonely individuals had measurably different patterns of gene expression in immune cells compared to socially connected individuals. Specifically, genes associated with inflammation were upregulated, and genes associated with antiviral responses were downregulated. The lonely individuals had immune systems that were primed for bacterial threats, consistent with the ancestral context of social isolation as exposure to physical danger, but less well equipped to handle viral infections. This gene expression difference was not small. It was found consistently and was associated with the subjective experience of loneliness independent of objective social contact, meaning that it was the felt sense of disconnection, not merely the number of social interactions, that drove the biological response. The cognitive consequences of chronic loneliness are among the most significant findings in this research area, and they operate through several mechanisms simultaneously. Sleep is the first casualty. Lonely individuals consistently show worse sleep quality than socially connected individuals. More fragmented sleep, less slow-wave sleep, more frequent waking. The mechanism involves the same hypervigilance discussed above. A brain in a state of perceived social threat does not fully disengage from threat monitoring during sleep. The result is shallower, less restorative sleep that compounds across time into cumulative cognitive impairment. The hippocampus, the brain region central to memory formation and spatial navigation, shows accelerated volume loss in chronically lonely individuals. A study following older adults over several years found that those who reported chronic loneliness showed significantly faster hippocampal atrophy compared to those who reported adequate social connection. The effect was independent of depression, physical health, and other confounding variables. The social disconnection itself was associated with structural brain changes. The dementia risk finding is the most striking. A meta-analysis published in Aging Research Reviews, synthesizing data from multiple large prospective studies, found that chronic loneliness was associated with a 50% increase in the risk of developing dementia. The effect size is comparable to well-established dementia risk factors, including physical inactivity, hearing loss, and hypertension. The proposed mechanisms include the cortisol-driven hippocampal damage, the sleep disruption and its effects on amyloid clearance, the inflammatory state that impairs microglial function, and the reduced cognitive engagement that comes from insufficient social stimulation. All of these operate simultaneously in the chronically lonely person, and they compound over the years that chronic loneliness persists. The quality versus quantity distinction is one of the most practically important findings in the social connection literature because it changes what an effective intervention looks like. Most people, when they think about loneliness, think about the number of social contacts, the size of the social network, the frequency of social interaction. More friends, more contact, less loneliness. The research does not support this as the primary variable. What predicts the biological and psychological outcomes associated with loneliness is not the quantity of social interaction, but the quality. Specifically, the degree to which social relationships involve genuine mutual understanding, emotional reciprocity, and a sense of being known and valued by others. A person with a large social network but relationships that feel superficial or transactional can experience the same biological state of loneliness as someone with objectively fewer contacts. Conversely, a person with a small number of close, deep relationships can have robust biological markers of social connection, even if their social calendar is limited. This finding is practically significant because it means that addressing loneliness is not primarily about increasing the volume of social contact. It is about the depth and quality of existing connections, which is a different problem requiring a different approach. It also explains why loneliness has become more prevalent in societies with high levels of social media use, despite the apparent increase in the volume of social contact that platforms provide. Frequent, low-depth digital interaction does not produce the neurobiological response associated with genuine social connection. The brain's social connection system evolved in the context of face-to-face interaction involving physical presence, vocal tone, eye contact, and touch, the cues that are absent or attenuated in digital communication. Here's what the research supports practically. The most effective single predictor of subjective social connection in the research literature is not how many people you know, but whether you have at least one relationship in which you feel genuinely understood. Not validated, not liked, but understood, in which the other person has an accurate model of who you are and responds to you accordingly. The presence of even one such relationship significantly buffers against the biological consequences of reduced broader social contact. Physical presence matters in ways that digital contact does not fully replicate. The oxytocin response, the hormonal counterpart to the cortisol response associated with loneliness, is most reliably activated by physical contact, eye contact, and the synchronization of breathing and movement that occurs in shared physical space. Prioritizing in-person interaction, even at lower frequency than digital contact, produces more of the neurobiological response associated with genuine connection. Contribution and purpose within a social context, being needed, being useful, having a role that matters to others, activates social connection circuitry that passive social consumption does not. This is why volunteering, mentorship, and participation in communities organized around shared purpose consistently shows stronger effects on loneliness outcomes than social activities organized purely around entertainment or recreation. And addressing the sleep disruption associated with loneliness through the same interventions that improve sleep in any context breaks one of the primary mechanisms through which loneliness accelerates cognitive decline. The relationship between loneliness and sleep is bidirectional. Better sleep reduces the hypervigilance that chronic loneliness produces. Reduced hypervigilance improves sleep. Interrupting the cycle at the sleep end is accessible and produces effects that extend beyond sleep itself. Here is the synthesis. Loneliness is not a soft problem. It is a biological state with hard consequences. Measurable changes in gene expression, cortisol levels, brain structure, immune function, and dementia risk that operate through the same physiological pathways as other recognized health risk factors. The evolutionary logic is clear. Social connection was survival critical. The brain treats its absence as a threat, and sustained threat activation produces sustained physiological consequences regardless of whether the threat is physical or social. What this means practically is that social connection is not a reward for getting everything else right. It is a biological need with the same status as sleep, nutrition, and movement. Treating it as optional or as something to address after more pressing concerns misunderstands what the research has established about its role in health. One deep relationship matters more than many shallow ones. Physical presence matters more than digital contact. Being needed matters as much as being liked. And the brain's response to genuine connection, reduced cortisol, reduced inflammation, better sleep, improved cognitive function, is as real and as measurable as its response to any other health intervention. If this changed how you think about loneliness as something more than a feeling, hit the like button.